Hi everyone, and a very warm welcome to today's APPS seminar series. I'm Kylie Chambers and I am from the Department of Primary Industries and Regional Development based in Northern Western Australia. I'm also the APPS Regional Counsellor for Western Australia and I'm hosting today's session. I would like to acknowledge the Balladong and Whadjuk people who are the traditional custodians of this land. I would also like to pay my respects to the elders, both past, present, and emerging, of the Noongar Nation and extend that respect to other Aboriginal people present. Today's seminar will be recorded and uploaded onto YouTube for later use. I would also like to note, before we start today's seminar, that there are some APPS student and early career bursaries available for the upcoming Australasian Soil-Borne Diseases Symposium, which close on the 1st of July, and the Australasian Plant Virology Workshop, where bursaries will be closing on the 1st of August. Today we have two fantastic presentations, and with great pleasure I would like to introduce our two speakers for today. Our first speaker is Sarah Nolan, who is a field plant pathologist at DPIRD based in Kununurra in far northeast of Western Australia. This region is home to the Ord River Irrigation Area, a unique tropical irrigation farming region with an interesting history and emerging industries, including cotton. Sarah is currently involved in establishing local disease diagnostic capability for WA cotton growers through the Australian Cotton Disease Collaboration. Her presentation will explore the realities of working in plant pathology in remote Northern Australia, including the rewards, challenges and opportunities that come with supporting agriculture at the edge of Australia's production systems. Our second speaker will be Dr. Viet-Cuong Han, who is a plant pathologist and microbiome scientist based at Curtin University. Soil and atmospheric microbial communities are often studied separately, yet they can form interconnected systems that may influence plant health across multiple scales. Today, Kung will discuss his recent work exploring how soil and airborne microbial communities contribute to plant disease suppression and pathogen exposure. So without further delay, let's get started, and I would like to invite our first speaker, Sarah. Whenever you're ready, Sarah.
Thank you. Well, good morning, everybody. My name is Sarah Nolan Gorman, and I am a research scientist with DPIRD in WA. And I've been living and working on Viet-Cuong Han country in the Kimberley region for the past 7 years. Before I get started, I just want to acknowledge and pay my respects to the traditional owners of this land, the Viet-Cuong Han people. So many people wonder why Kununurra. Well, I grew up on the Darling Downs in Queensland and studied applied science agronomy at the University of Queensland. And when I graduated, I was just looking for sort of any opportunities in agriculture that I could find. But what actually led me to looking for work in Kununurra was a photo. During uni, I'd worked for Pacific Seeds, and a colleague came back from the production farm that they have up here with a picture of a sorghum crop just ready for harvest with these amazing red ranges in the background, which you can see in this photo on the slide here. And that was enough. I had packed my bags basically and was ready to go. I moved up in 2019 thinking that I'd stay for a couple of years, and I'm still here 7 years later. Locals say that you drink the water and that just happens, so it's a pretty common story. After arriving in Kununurra, I spent my first few years working in cotton agronomy trials, but there up came an opportunity to work in or to move into plant pathology research. A role came up and I was just really excited by the chance to learn something new. So just for context, to understand what it means to do plant pathology in Kununurra, you kind of need to understand where Kununurra actually is. So the Ord River Irrigation Area sits in the far northeast of Western Australia in the Kimberley. So we're roughly about 3,000 kilometres from Perth and actually far closer to Darwin than to any of WA's other agricultural regions or cropping regions, you could say. And for context, it's only an extra 100 k's for me to get back to Dalby in southeast Queensland than it is to get to Perth, the capital. So the Ord could kind of be described as an agricultural island, but not necessarily just because we're surrounded by water. We do have the Ord River that feeds our irrigation system and Lake Argyle. During the wet season, it feels like everything is just water, but also because we're isolated geographically, socially, and agriculturally. The Ord was developed following the construction of the Ord River Dam and later Lake Argyle, which you can see photos on this slide of both of those. So it was transformed from what was previously pastoral cattle grazing country into a large-scale irrigated farming region. So we grow mangoes, cucurbits, maize, chickpeas, seed crops, and then more recently cotton. The soils are self-mulching clays across the floodplain, and there is a gravity-fed open canal irrigation system that distributes water across the farms. We have a distinct wet and dry season, which means we can really only reliably grow one crop per year because the clay and then all of the rain limits the trafficability of the paddocks for a few months of the year, really. What makes the Ord particularly interesting from a plant pathology perspective is that it operates really differently to most of Australia's traditional farming systems. It's a contained landscape surrounded by untouched rangeland. I was hoping to point at where we are. So here you can kind of see on the presentation that is the Ord River irrigation area. It spans up here to the north, closer to the Northern Territory state border, and is going to expand further out this way as well. And all of this is just rangelands. So it creates a real opportunity, one, both from a biosecurity risk. We're a border town, we're close to Southeast Asia, and also an opportunity because we're a contained region, and if you can coordinate and sort of be prepared, you can do landscape scale surveillance and disease management in a way that a lot of other farming systems can't. They don't have that same opportunity. The island nature of the Ord has shaped the region's agricultural history in some pretty dramatic ways. The Ord has lived many lives as pest diseases and the environment have repeatedly shaped and ended its industries. So in the 70s, a native lepidopteran species developed insecticide resistance and collapsed the cotton industry. Rice blast, suspected to have arrived with migratory birds, sort of ended the rice industry. Sugarcane smut impacted the sugar industry. And today we have a sort of specialized chickpea, Kabuli chickpea variety, the Kimberley Large, which goes, it's quite a big seed and it goes into a specialized high-value horticulture market, and it is limited also by its intolerance of disease pressure and waterlogging. So historically here, the disease doesn't really just affect individual paddocks, but kind of has just repeatedly determined which industries stand the test of time. So after moving into the plant pathology role, when I first began working as a field pathologist, there hadn't been one working in Kununurra for decades. Most of my colleagues were 3,000 k's away in Perth. There wasn't really a project waiting for me, not really any local diagnostic records. And although there was a lot of inexperienced people, there wasn't really anyone that had worked in crop disease. But there was one of my colleagues, Andrew Taylor in Bunbury, who has fielded and continues to field many phone calls from me about things. My position came about because growers wanted local pathology support. So I spent most of my initial first days on the job talking to growers and trying to understand what was happening. And I just very quickly found out that there is just like not really a guidebook to this kind of thing when you first start in the role, and there's nothing there to rely on, not a lot of certainty. Early on, I spent a lot of time finding what I thought were diseases in crops, only to discover it was really just something else. I once found a crop of biofumigant turnips with what looked to be a wilting problem. Looked pretty sweet, thought that was disease for sure, and I dug it up and it turned out to be root maggots, which was just truly so disgusting. It was not a pathogen. I found really strange patterns in the paddocks that I thought, great, this is absolutely some soil-borne disease. That turned out to just be low spots in paddock waterlogging that needed to be re-laser leveled. So that's a pretty easy fix, really. The region itself doesn't have a detailed diagnostic history. So a lot of the time it's very much Is it a plant disease on a decision tree? It's almost like everything leads to something else before it could really be considered a disease, unless it's obvious. One of my favorite examples of this is from early in my time here. We were trialing a crop called Plantago ovata, which is the plant that produces psyllium husk on the research station. And some of the plants were sort of producing like really low spots. The growth was a bit constrained. The seed or the flowering heads were browning off and the flowers were sort of dying. And we ran through a range of possibilities, but one really serious hypothesis we considered was that bush turkeys had been sleeping in the crop and had somehow vectored a disease. You could kind of see, like, aside from my colleague here sleeping on the Plantago, but other big dips where it looked like something had been lying down. And while it sounds a bit random, it actually isn't really that far-fetched, because rice blast has long suspected to have arrived in the Ord via the migratory magpie geese that we get here annually that come from Southeast Asia and come to roost and breed in Kununurra. We never really resolved that issue, couldn't prove it to be the plains turkeys, but what came out of the process of that and a lot of these sort of situations of not really being sure what was going on was that there was a lot of connections and talking to people and trying to figure out what other people and what other professionals thought of the situation. And so from that, we get NARCs come to survey every year here in Kununurra, the Northern Australian Quarantine Strategy. And through meeting them in the paddock and noticing that there, was sort of a mutual beneficial need for me to learn a little bit more, have a bit more on-the-job training, and they actually needed a plant pathologist at the time. It resulted in a short secondment with them in their Darwin plant pathology team. So I worked in the Darwin lab. I attended plant health surveys around Darwin and the Torres Strait, and just met a whole community of plant pathologists working in Northern Australia and facing sort of similar issues to me. They introduced me to the concept of the pressure cooker autoclave, which was a lab staple in the first few years that I didn't have any lab equipment or really even a lab. So with the NARCs, obviously they're biosecurity focused, and the overlap between agriculture and biosecurity is something that you're always aware of up here and in the north. We're really close to Southeast Asia. You can see here that Timor is really only about 850 k's away as the crow flies. The monsoon brings northerly winds that can blow in insects, and that might be vectoring viruses across the Timor Sea, and a lot of high priority plant pests are in those regions. And, we're also sort of close to this Darwin border here. So at some stage a few years ago, a suspect myrtle rust report had us needing to do a helicopter survey in the remote North Kimberley around here, this sort of zone. And we were hopscotching on the helicopter between Mound Springs, a sort of spring-fed endangered pockets of vegetation that dot across the mudflats in that region to try and hopefully find this myrtle rust that had been reported by the DBCA rangers. So in the Ord, the challenges have changed over time, but the underlying tension between developing agriculture and then responding to the environment in a place sense and then also in an environmental sense has been a part of the story of the Ord from the beginning. So this sort of brings us back to cotton, which is where we're at now. And it sort of starts to feel pretty relevant to the present. With the release of Bulgard 3 in 2019, cotton came back to the Ord with a much better toolkit for dealing with the tropical lepidopteran pest that ended the first cotton era, and actually a couple of eras since then before Bulgard 3. A ginning facility opened last year, and cotton is now the crop underpinning the agricultural industry in the region. Given how rapidly it's expanding and given the plant disease issues that challenge cotton production in the eastern states, building local cotton pathology capability in Kununurra has actually become genuinely important for us. So through the Australian Cotton Disease Collaboration, DPIRD has a project that I lead in Kununurra to build capability for local pathology support for WA cotton growers. So we're just working to get ahead of the disease risk as the industry continues to expand. The project involves disease surveys across commercial cotton fields, diagnostic support for growers, and also data contribution to national surveillance efforts. We're increasingly able to isolate pathogens, extract DNA, and perform PCR right here in Kununurra. This is me and my colleague Kim in our brand new molecular lab here. So it sounds pretty basic, but it's a huge game changer when the alternative is a round trip to Perth with the samples. So from our surveys in the 2025 season, we've found distinct differences in fungal pathogen communities between the upper and lower canopy of our local crops, which is quite different from the known knowledge about these complexes from the eastern states. We found alternaria leaf spot complex dominating up the top, and target spot was more common below. We consistently found quite low but repetitive levels of boll disease across the region. You can see here on this graph that everything is really quite low. We went through and counted all the bolls actually on these plants to figure out how many of them had any kind of boll disease. So and from this, we found Fusarium, Lasiodiploidea, and Alternaria were the main pathogens associated with the boll diseases and sometimes co-infected the same boll. So the disease pressure so far is low and manageable, but there is growing industry momentum to support the new gin by planting back-to-back cotton seasons while more agricultural land is developed for scale to be able to support the success of the gin and the investment into the infrastructure, which of course does slowly become a more conducive system for increased disease pressure. So just finally, the order's been a place where it seems like the natural world seems genuinely determined to shake off whatever agricultural system tries to tame it. Plagues, resistance, floods, weeds, pests, and diseases have all had a go. It has, as an early career plant pathologist, just been a really wonderful place to learn about connections and understanding history to support sustainable development. It's an amazing place to work. Inspecting crops with the ranges and the waterfalls visible from the paddock has just been so cool. The work really connects to the natural world, agriculture, biosecurity, and history in a way that I never expected when I was just in Toowoomba working at Pacific Seeds and looking at a photo of a sorghum crop with the ranges behind that I would like to visit. So I guess my final point is go north, go west, go out. Thank you. Thank you, everyone.
Thanks, Sarah. If anyone has any questions for Sarah, if you could either unmute and ask or add it to the chat box and I can read them out for Sarah. Well, I guess while we're waiting for people to add any questions into the chat function, it sounds like you cover a lot of different hosts, a lot of different diseases, and a lot of area. Is a lot of your stuff going out on farm and covering that whole area by just traveling long distances or getting sent in samples or a combination of the two? And where do you
Yeah.
kind of get the resources to when you're unsure of something?
Well, it's a big area, but it's quite contained. So we are able to sort of, if we get a call about something, we'll drive out there and have a look. Our surveys we're able to sort of go through the day and then drive back to the lab for processing. So yeah, it's a lot of us just sort of driving around and looking at things, Kim and I. And then in terms of finding the resources to figure out what people are, that ACDC, our project collaborators in Queensland, where there are cotton industries, has been really valuable. And when it's something random, I just tend to go, I feel like over the period of my career, I've managed to make connections with people that know about different things. And so I just go straight to the person who I think might know about it. If it's something on a banana or in sunflowers or mangoes, whatever it might be, it really, like for me, it really has just been all about the connection to different experts.
Rebecca's just asked how much area is cotton currently cropped in Kununurra?
In our system, I think we have about 20,000 cultivated hectares, and around 15,000 of those is cotton now, between 10 and 15, depending on the season.
And Kith's just asked if you have ever seen any angular leaf spot.
Yes. Yeah, we do have that occasionally. We get Ramularia grey mold here that has an angular leaf spot.
Is there any other questions for Sarah? Any serious soil-borne diseases of cotton?
There are a lot of
Wadiya, sorry.
No, you're right. There's a lot of serious soil-borne diseases. They're really common on the east coast. There's verticillium, fusarium, reoccurring wilt, and black root rot. But we haven't really seen much here in the early seasons. If so, we plant at the beginning of the wet season or the end of the wet season in January. So we do tend to get a lot of rain through that period as well. And we do see sometimes in the tail drains of the paddock where it's a bit lower, there might be some die back a little bit there, but it's not really, it's more environmental than it is like a really strong pathogen. So it might be charcoal rot or Pythium phytophthora might come in at that time.
And the final question from Andrew. Sorry, I'm probably going to butcher the pronunciation of this. Have you looked at the native Gossypium to see what they host?
There, we have not done that. That is part of a project in the ACDC. There has been some work looking into whether the native Gossypiums will host different viruses and whether they might be reservoirs of viruses. But at this stage, we're sort of more focusing on what is going on in the crops, but it might be an avenue later on.
Well, thank you, Sarah, for your time. And I will move on to our next speaker, who is Viet-Cuong Han. So if you could share your screen, Kung, and whenever you're ready.
Yes. Thanks, Kylie, for inviting me to this seminar. Also, thanks, Sarah, for a summary of the plant pathology research and what's happening in the northern Australia with plant pathogens. And my talk today will be about soil and air microbiomes and their implications for disease suppression and pathogen exposure. Yeah, I'm Kuang from Curtin University. Yes, just a brief statement about my academic journey, which spans from Vietnam and then South Korea, and recently in Perth in Western Australia at Curtin University for my PhD. My main research centers around plant pathology and biological control and microbiome-based management of plant disease. And yeah, I will acknowledge my project team members and colleagues for the project I was involved in and for the work that I will be presenting in this presentation. The first one for soil microbiome from my PhD project and follow up by the air microbiome after my PhD. And yeah, let's get started. So as you all probably know about the plant disease triangle, yeah. So originally, the researchers proposed plant disease triangle to emphasize that there is a three criteria: host, pathogen, and environment. When they meet together, the disease will occur. But nowadays, with the regard for the microbiota, they consider pathogen as just a component of the whole microbiota, which contain both uh, uh, pathogenic microbes, beneficial microbes, and other microorganisms. And rather than trying to get plant disease, now the triangle to focus on plant health. This emphasizes the critical role of microbiomes in the plant health and plant protection. So yeah, what makes up the microbiota? Yeah, the microbiota contain, as I said, contain different types of microbial groups, which contain fungi, humices, bacteria, nematodes, protozoa, and viruses and other microorganisms. And the number of the genes of the micro microbes exceed the number of genes of the plant. This emphasizes that microbiodiversity plays an important role in the plant health as well as for the suppression of the plant pathogens. So, yes, one of the most greatest example of how soil microbes can help plant to uh, defend the pathogen infection or protect the plants from pathogen infection. It is disease suppressive soil. Disease suppressive soil were defined as where the pathogen failed to establish or cause minimal disease, or the pathogen or the disease decline over the time. And the disease suppressive soil is mainly mediated by microbiome. That's why we call microbiome driven pathogen suppression. And in my PhD project, we focus on the Scartinia, which cause the Scartinia stem rot in different range of the plants, especially in broad arc plants such as canola, also and legume as well. And the disease cycle for for the pathogen is everything starts from the sclerotia. This is the fungal structures in the soil, and the infection starts from the sclerotia germination. And there's two types of germination. And the first one, that sclerotia can germinate mycegenically to infect the stem base. And the other main pathway, that sclerotia can germinate carpogenically to apothecia. Is like the apothecia will then have the ascospore to travel around to infect the above ground plant parts such as the stem and the leaf. Yeah, that's the two main pathway for these pathogens. So the key idea from my PhD project, whether the soil microbes can suppress the infection of Sclerotia and also can inhibit the germination of Sclerotia. If the Sclerotia can be, if the Sclerotia germination can be inhibited, there would be no source of infection for plant. That's the key point for my project. That's why we focus on and try to understand the suppressive soil microbiomes against Scartinia scarsierum. And to do that, we first sought out to find where the suppressive soil against Scartinia scarsierum can be occurred. And we try different area with different type climates and different rainfall level across Southwest, across the wheat belt in West Australia, and we finally ending up with a field from Kirchenap in West Australia, as this field grow with canola and rotated with different legume and cereal crops, and it has the important point is that it has the long history of very low Scartinia stem rot disease. And in 2016, as you can see here, and it has very severe Scartinia stem rot, but later the disease seemed to be less important. That's very good signal of disease suppressive soil. And we compare with the nearby paddock, which also experience pretty the same rotation system, also contain canola, lupin, legume, and cereal. But this nearby field has very severe, very high disease of Scartinia year and years. That we call that's very conducive field, a conducive soil. And we compare this one with the natural control as well. They own very closely to each other. And this is based on the farmer discussion, the history of the field and the management as well. We got this, and to validate that, we collected the soil samples for bioassays and for biological and chemical analysis later as well. For first, we performed the bioassays to confirm whether what we saw in the field, for example, the suppressive soil, are actually suppressive. That's why, and then we perform a bioassay for the infection, basal infection in the plants in the lab, in canola plant. And as you can see here, this is suppressive soil. I mean, has very low AUDPS. It kind of has a very low disease progression. It means that suppressive soil can suppress the infection of Sclerotinia into the plants that we validate in the lab. And compared to that, the conducive soil show very significantly higher disease, and especially for the remnant vegetation where no host plant of Sclerotinia were grown before, but it shows very, very high disease progression. It means that this is very susceptible to the disease, and I will show why remnant vegetation soils are very susceptible. But generally, suppressive soil shows suppressiveness against Sclerotinia. So to further confirm whether the suppressiveness is mediated by microbiota or not, we perform another bioassay. And when we mix the conducive soil with suppressive soil, it shows an increase in the suppressiveness of the conducive soil rather than conducive soil alone. This means that the suppressive soil can be transferable to the conducive soil. And again, when we did the heat treatment at 80 degrees of the suppressive soils, it caused the loss of disease suppression. Taken together, this indicates that the suppressive soil we collected are mediated by soil microbiota and can be transferable to conducive soils. And this is, as I mentioned, the fungal pathogen Sclerotinia sclerotium has two main pathways for infection. Now, the first one, we try to target the inhibit of the basal infection. And the second pathway is that the sclerotia can be germinated carbogenically to apothecia. That's the main pathway. Now we try to see whether the suppressive soils can inhibit the germination of sclerotia carbogenically. And actually, the suppressive soil did. As you can see here, it shows a strong suppression of sclerotia, resulting in very low germination of sclerotia compared to conducive soil. Once again, remnant vegetation is so very susceptible to Sclerotinia sclerotia germination. So we ask what makes up the microbiota of the suppressive soil, and we did the high-purpose sequencing targeting the bacteria, fungi, and eukaryotes. But in terms of this presentation, I will just show the data on bacteria. And we saw the significantly different pattern in bacteria between suppressive soil and conducive soil. As you can see here, we get more amplified sequence variance in suppressive soil compared to conducive soil. However, when we perform the diversity, when we calculate the diversity based on the richness and the evenness, there's no difference. This indicates that the suppressive soil was possibly dominated by a few bacteria groups. And here we perform another analysis to see what the bacteria community in the soils. And as you can see here in the suppressive soil, notably we saw two general bacillus and neo-bacillus seems to be higher abundant than that in conducive soil. And as you can see here in remnant vegetation, where they are very susceptible to the disease, there's no presence of bacillus and neo-bacillus. That's the point. With this data, we further focus on trying to figure out what is the drivers of the soil suppressiveness. And we performed the random forest machine learning as well as linear discriminant analysis effect size to detect and identify the biomarkers or the potential drivers that lead to the soil suppressiveness. And as you can see, once again, these both analyses suggest bacillus and neo-bacillus as the potential drivers of the soil suppressiveness. And to further validate what we found, we performed the net-shift analysis. This one can suggest what is behind the disease suppression in the soil suppressiveness. Once again, bacillus was indicated that the key driver for the soil suppressiveness. And also, we performed the microbial co-occurrence network to investigate the bacteria interactions between the bacteria in the community. And as you can see here in the suppressive soil, the network is much more condensed and more connected and bigger network. Especially, this network is highly centralized compared to network in conducive and rampant vegetation soil. As you can see here, this is highly centralized, and the microbes in this hub may play a key role for regulating the whole bacteria community in suppressive soil. That's why when we did the heat treatment before, it caused a loss, maybe it caused a collapse of this hub. That's why it caused a loss of the suppressiveness in a suppressive soil. Yeah, and among these hub, bacillus still remain one of the key node in this network. And yeah, that's what we got from the high throughput sequencing data. But now to validate what we got, then we performed the validation experiment in the lab. We did the isolation of bacteria, and we got over 200 bacteria isolated from 3 different soils. And here I show and in the suppressive soil, we got 62 bacteria isolates that show the suppression against Sclerotinia in vitro in the plate testing. And this number is double higher than that in the conducive soil. It means that more high number of antagonistic bacteria in suppressive soil. That makes sense. It may result in the higher suppression. And among all of these, yeah, we did Sanger sequencing as well to identify exactly what they are, and all of them belong to Bacillus and Streptomyces general. And some of them are some of them are possibly as a new species that hasn't been reported before against Sclerotinia. And this is in vitro. So to validate what we got from in vitro, we performed the bioassay in the plant, in the seedlings. Yeah, we introduced bacteria again to the soil to see whether they can suppress the basal infection by Sclerotinia, and we select 3 bacillus and 3 streptomyces isolates, and we perform them either in isolation or in the mixture, and all of the single strain or a mixture of the strain can suppress can suppress Sclerotinia infection. Yeah, that's good news, and especially when we put 3 bacillus strain in the mixture, it shows the strongest inhibition, significantly higher than the disease suppression efficiency from single strain. This indicates that maybe 3 bacillus strain can result in the synergistic action. That's good news, and that's what we got. And for the soil suppressiveness. And yeah, the next one is in the soil microbial community, which not only contain the beneficial or antagonistic microbes that can suppress the plant pathogens, the soil can be a can harbor the pathogens and the hidden pathogen or the latent pathogen as well. That's why from all of the 3 soils I mentioned before, we investigate the fungal community in the soil. And as you can see here in the disease conducive soil, there is significantly higher abundance of plant pathogens than that in the suppressive and remnant soil. Yeah, this makes sense. And yeah, this soil is very susceptible to Sclerotinia before. So yeah, this is for the traffic mode of functionality of the fungal community in the soil. So we zoom in to see what exactly fungal pathogen present. Especially we investigate both the fungal pathogen of the plant, but also the humans and animal as well. We detect all of them in the soil, but here I just show the data on fungal pathogen we detected. As you can see here, as conducive and suppressive soil are rotated with different type of plant from brassica, legume, and cereal. Yeah, that's why we got most of the main pathogen of these crops in the soil. But the important thing here is that the blackleg, one of the most important pathogen of canola. But yeah, as you can see here, we got in conducive and suppressive soil. That makes sense, but we still got that in rampant vegetation in the nearby area where no host plant were grown before. And the same thing for Sclerotinia. And yeah, this indicates that maybe the pathogen or the spore or the mycelia of this pathogen can travel to the rampant vegetation from their field. And importantly, we didn't detect the known pathogen of the known crops, but we detect some of the pathogen of the crops that maybe haven't been grown on around that area before, such as the pathogen of the noto ginseng, strawberry, and wasabi. Yeah, these taken together, these emphasize the potential of using the environmental DNA or high-throughput sequencing for detection of the fungal plant pathogens. And building up on what we found of the fungal community and fungal pathogen in the soil, we recently developed kind of a new mobile eDNA platform, environmental DNA platform for monitoring microbiome community across landscape scales. And this is what we did. And because we asked the question, we detected pathogen in the soil, whether they can be in the air. Yeah, that's the key question that we asked. And we designed the airborne eDNA sampling platform like this. We put the air filter in both the door of the car, and we travel around the southwest Western Australia across over 1,000 kilometers, and it came across different bioregion where different vegetation or plants were grown. As you can see here, yeah, this may allow us to monitor the landscape microbial community across Western Australia. And yeah, what we found, which is the same with what we got in the soil. As you can see here, in the air, we still once again detected different fungal pathogens of the agricultural crops, forestry, of the ornamental plant as well. And as you can see here, some of these pathogens were detected in the soil before. Now we got in the air. This suggests the interconnected uh, system between the pathogen. They can be in the air, also in the soil. And importantly, we also detected one of the most important pathogen of the cereal crops, such as barley, Peridophylla teres. Yeah, it has two different forms, such as maculata and teres. And this species is known for developing fungicide resistance. Yeah, it's very important point. That's why we try to see whether from our data, from our sample, we can detect the fungicide resistant mutation. Yeah, it actually, we did. Yeah, we detect the mutation targeting the one of the CYP51 gene of the for fungicide resistant. And here we got half for the sample here. From sample 5 here, we got half of them were has the moderate to some with highly resistant, and from the form teres, we got most of them are moderate or highly resistant to fungicides. Yeah, taken together, this emphasize that our mobile eDNA can allow us to not characterize the microbial community, detecting pathogen, but also identify the resistant potential. Yeah, taken together, my talk was about the soil and air microbiome, which can form an interconnected system through the persistence and dispersal. And soil microbiomes can drive the disease suppression, acting as both reservoirs for antagonistic bacteria and antagonistic microbes and latent pathogens. And finally, mobile eDNA can enable us to landscape scale detection of hidden pathogens and resistance. Yeah, as you can see here, and we detect a lot of fungal pathogen or different pathogen, but because we use high throughput sequencing data, it just detect the DNA of the pathogen that's shed in the environment. We don't know of it didn't confirm whether the pathogen are viable or alive. Yeah, that's one of the limitations of the eDNA. But generally, eDNA can allow us to characterize and monitor the surveillance or the presence of the pathogen in the environment. Yeah, and yeah, thanks everyone for listening, and also thanks to my supervisor and collaborators as well for all the work I show in this presentation. Thank you very much.
Thanks, Kung. Just to keep us on schedule today, if there is a quick question for Kung, we might only have enough time for one, and there's one already in the chat here. Disease suppressiveness is often emerging from complex microbial communities involving hundreds of interacting bacterial and fungal taxa. How do you reconcile the activity of a few isolated bacillus strains on agar with the greater complexity of the disease suppression in field conditions?
Yeah, thanks for that question. Yeah, it's actually that experiment is kind of validation of what we found in high throughput sequencing data. Yeah, because as I showed before, we analyze the data, and it suggests that bacillus as one of the key driver for soil suppressiveness. That's why we follow up by isolation of bacillus and try to validate whether they actually suppress the disease or not. Yeah, that we try to mimic what happened in the field, but yeah, and finally we indicate that bacillus can be served as the key driver for soil suppressiveness and also as the maybe the indicator for the soil suppressiveness. And that's the implication, potential implication from our finding. But yeah, as you know, that is a soil, that's a very complex interaction. Also when you perform experiment with single strain or consortium of different strain or different species, but sometimes it couldn't mimic exactly what happened in the soil. But yeah, that still suggests a future research which is needed to understand the more complex. Thanks for that question.
Thanks, Kung. So that brings us to the end of today's seminar, and I just want to extend a massive thank you to both of our speakers, Sarah and Kung, for sharing their research, and for all of you for your active participation and attending this seminar. The next seminar will be held on the 14th of July and hosted by Sue Pederick, who is the South Australian Regional Councillor and the APPS Region Council Coordinator. I'd also just like to reiterate that there are the APPS student and early career bursaries available for the upcoming Australasian Soil-Borne Disease Symposium and the Australasian Plant Virology Workshop. Thank you.
Thanks, Kelly. Thanks, everyone.
Thank you.